Hello and welcome to the Microbiome Medics podcast. I'm Dr. Siobhan McCormack.
And I'm Dr. Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science, and we have spent the last 5 years deep diving into the world of the gut microbiome.
We've been analysing the evidence, appraising the studies, speaking to the experts, and writing the Gut Microbiome for Clinicians e-learning course for the BSLM Learning Academy.
We have discovered that the gut and other human microbiomes are pivotal to every aspect of your health and physiology, and the pillars of lifestyle medicine hold the key.
So in these Microbiome Medic podcasts, we'll be covering the basics, bringing you the latest research and talking to the experts on all matters microbiome.
We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients.
Hello and welcome to this month's Microbiome Medics podcast. Today we're going to discuss the leaky gut. What is it? Why should we know about it? And we'll also be tackling a little bit of the bad press about this condition and taking a deeper dive into its relationship with disease. So why are we doing this? Well, there was an article in the British Journal of General Practice in December 22 by McCartney et al. And they cited leaky gut as pseudoscience. Now, they also called it a demonstrably incorrect diagnosis, which is quite interesting. So we're going to talk about this and decide whether or not those statements are correct about leaky gut. And if not, then we'll try and correct some of those statements. Okay, so Shiv, what is leaky gut and why is it so dismissed by clinicians like Dr. McCartney?
Hi, Sheena. Well, I think we have to agree that the term leaky gut doesn't sound very scientific. It's, ubiquitous in men's and women's health magazines, and everywhere you look on TV and radio, people are talking about gut health and leaky gut, and it is one of those things that make lots of my GP colleagues kind of groan, and I think it's because it's an area that we weren't taught about in medical school. The actual scientific term is increased intestinal permeability, but that doesn't run off the tongue quite so easily. So I think we have to be aware that this is being talked about a lot in the media, and that our patients are going to know about this term and will be asking us about it. So I think the first thing before we sort of do a deep dive into what it is, how we diagnose it, I think this whole area of gut health is quite confusing for clinicians. And Sheena, you and I have spent the last 3 to 4 years deep diving into this subject, and we've read numerous scientific papers about the gut microbiome, and certainly increased intestinal permeability is frequently cited, and they often put in inverted commas, aka also known as leaky gut. So they are starting to use this term as well. And in translational science, we're trying to sort of appeal to the general public. It seems to be a term that they like and that they kind of understand and are drawn to. So I think we have to be aware of that. But before we get down to exactly what it is and how we diagnose it, I think it's really important to understand gut health and its importance. And the way to do that is to look at the gut barrier, which I've kind of fallen in love with really over the last few years. So Sheena, if you imagine yourself as a tube and you have interfaces with the outside world, so you're trying to survive in this complex environment. The outside of your tube is your skin, so that's about 2 meters squared. The inside of your tube is actually your gut. People don't see that as an interface between themselves and the outside environment, but it is. It's open to the outside world. Now, that is from your mouth to your anus, and it's about the size, the surface area is about 32 meters squared, which is apparently about the size of half a badminton court. Now, that is a massive area that you have got to guard 24/7 against toxins and bacteria pathogens and any harmful molecules that you may present themselves to you. So if we think about this as a kind of highly engineered passport control, and that explains why 70% of our immune system is located in the gut. That's a pretty astonishing statistic. And it sits in this gut-associated lymphoid tissue all along the gut wall. And when you think about the massive area that your body has to patrol, that kind of then begins to make sense. The other thing that's key to understand when we're talking about intestinal permeability is your gut wall is meant to be permeable. It's a physiological necessity because you have to allow nutrients and bioactives and water through. But at the same time, it's got this really delicate, complex balancing act because it's also got to deter and block potentially dangerous molecules, such as parts of bacteria or various toxins or antigenic food particles. So it's really constantly testing and blocking some particles from admission and allowing other particles through. So this beautiful gut epithelium is a dynamic responsive structure. It opens and closes with various signaling molecules. Remarkably, Sheena, there's only one layer of cells between you and the outside world. So if you imagine this cobblestone structure of cells called, these epithelial cells are called enterocytes, and they kind of sit closely together, kind of shoulder to shoulder. But these cells can move apart. And the way they move apart is by opening up complex, tight junction structures. And there are several different types of junctions between cells. But if we just focus on one called the tight junctions, now these are made up of over 150 different types of proteins, and they open and close with different signaling molecules, one of which is something called zonulin, which we'll be coming back to later. And that causes these cells to move apart and allow things to go through, and then they close afterwards. So you can see that you've got these cells and these complex junctions. And then on top of that, we have mucus. So the mucus layer in the small intestine is only one layer thick. And as it moves down to the colon, the large intestine, you get these two layers of mucus. Now you get a thinner adherent layer, which is mostly no bacteria in it, and it's got to protect the gut wall from everything in the lumen. And then on top of that, you've got a wider, more fluid mucus layer, which is dotted with various bacteria and immunoglobulins, and also these wonderful antimicrobial peptides. So you can almost see it as a kind of sanitizer gel. Your bacteria squirt various peptides into it, and your cells, something called the Paneth cells, also secrete various antimicrobial peptides such as defensins, and these all act to deter microbial growth or pathogenic microbial growth. So, and then on top of that, you've got your layer of gut microbiota, so this wonderful layer of bacteria, mostly bacteria and archaea and other organisms. So you have this brilliant sort of bacterial protection layer as well. If we go the other side of your enterocytes and look down towards the body side, you have this area of lamina propria, which is dotted with huge numbers of immunocytes. So it's things like macrophages and dendritic cells and B cells and T cells. And then very briefly, you have this fantastic enteric nervous system, which is your gut nervous system, and also lots of hormones that the gut produces as well. So you can see that this amazing barrier structure is really quite complex and highly engineered. And any defects in one or several of these aspects of the gut barrier can allow the gut barrier to break down and allow the entry of potentially dangerous molecules, which then trigger the immune system and cause inflammation. So that is kind of what the increased permeability is about. It's about a pathological process which then damages the body in various ways. So, Sheena, if I can pass over to you now and sort of look at the things that maybe can damage the gut barrier.
Yeah, thanks, Shiv. Yeah, that was a really comprehensive explanation there. And I think, just shows the detail in the gut wall. It's not a simple thing. There's so much going on there, and likewise, the factors that can actually influence our gut barrier, they're not simple things. There's often sort of complex processes going on with these things. But just to sort of go through a couple of things that can actually affect your gut barrier. So, for example, if you eat a high-fat, high-sugar diet, you're going to encourage more of the wrong type of bacteria within the gut. And these sort of more pathogenic bacteria can actually eat the mucus and erode its protection. likewise, when these bacteria break down, the wall of the bacteria contains a substance called LPS, which, when it's broken down, can actually be quite pro-inflammatory, and it's an endotoxin. So that can be very damaging for the wall of the gut as well. So if we have too many of these really harmful bacteria in our system, then there's several ways that they can damage it. But they can also outcompete the very beneficial bacteria that are in there. So if there's too many of the harmful ones, they can knock out the beneficial bacteria. And the beneficial bacteria actually play a role in protecting our gut membrane and our gut wall. They help to produce mucus. They help to repair the wall. So if there's not enough of the very beneficial bacteria in there, then of course it's going to have a knock-on effect on that. So there's other things that can influence it. So drugs, for example, anti-inflammatories, we all know that they can be very damaging for the gut wall, but they can cause little erosions and perforations and ulcerations to the gut wall, all by damaging the sort of tight barriers that between the enterocytes, and in addition, other drugs like, for example, PPIs, omeprazole, lansoprazole, these type of antacids, we often give them alongside anti-inflammatory drugs to protect our gut, but in actual fact, it causes even more erosion of the gut wall when we give them alongside anti-inflammatories. And it increases, because it lowers the pH in the stomach and the small bowel, it actually allows more overgrowth of those harmful bacterial species in that area, which of course can then lead to more damage. So it has a sort of double whammy there. So that is actually a little bit concerning for me as a GP, because of course I see an awful lot of people on these medications. Other things that can damage the gut wall, well, infection. We know that harmful bacteria going in there and releasing endotoxins can actually be very harmful for the gut wall. Emulsifiers, I'm going to go on to talking about them in the research paper, but they can be very damaging to the gut wall. Bile acids, you may have heard of. So when we eat something fatty, we release bile acids, and bile acids are very protective to our gut wall. They're very anti-inflammatory, and they encourage the beneficial bacteria to flourish. However, if that system's not working effectively and we don't produce enough of these bile acids, then we can get microbial dysbiosis, so an imbalance of the gut bacteria, and we can get leaky gut there as well. And likewise, if we eat too much fatty foods and we have too much bile acid in our system, some of it's converted into secondary bile acids, and these can be toxic to our mucosal wall, and they actually can be quite carcinogenic as well. So that can be quite damaging to the gut wall. High sugar, I've talked about a little bit, but high sugar on its own, so if your sugar levels are very high, actually damages the tight junctions in the epithelial lining and encourages the overgrowth of harmful bacteria. And alcohol, of course, can be pretty harmful there as well. You get a lot of mucosal erosions and ulcerations with alcohol. And again, alcohol does encourage the wrong type of bacteria within the gut wall as well. People with obesity have higher levels of LPS, that harmful endotoxin, and more of the harmful bacteria, which will again erode that. So lots of ways that the gut wall can indeed be eroded from all sorts of things that we come into contact with in our modern Western world. So yeah, it can be damaged by a lot of different things. Moving on, Siobhan, we were going to talk about how we measure intestinal permeability. Have you heard of any intestinal permeability tests, Siobhan?
Well, certainly it's obviously reading the science literature. There are various tests they can do in the laboratory. So they can sort of test in cell tissues and biopsies, and also in animal models as well. It gets much more complicated when you move to humans and trying to test it. And certainly if you're looking at what's done, what's available on the market now, certainly NHS, apart from in research units, aren't doing a lot of testing, but you can get various wellness groups who are sort of interested in gut health will do various markers, but that's when the controversy starts, because it's very complex how you measure it, whether the markers are correct and whether they're sensitive and specific enough. And I think that's when a lot of the controversy comes as to whether it's a good test for leaky gut. The other problem is when you look at the kind of symptoms that people are presenting with, they're very, very vague. Tiredness, aches, joints, not feeling cognitively, bright enough, headaches. It's very, very vague symptoms. So it's actually quite difficult to sort of work out how to examine or how to test for these things. So what do you think, Sheena?
So there are a few tests, aren't there? I mean, the tests that I came across for this. So I think what is certainly been cited in the literature as more of a gold standard test is the mannitol lactulose test. So basically, mannitol and lactulose are carbohydrates, so sort of sugars, and they are different sized molecules. And so giving patients some oral mannitol and lactulose to ingest, and then measuring the quantity of mannitol and lactulose in the urine over a sort of 24-hour period. So you measure it sort of 1 to 2 hours, then sort of 7 to 8 hours, and then 24 hours later, you can get a ratio of lactulose and mannitol in the urine, and depending on that ratio, you can work out how much leaky gut there is, and the sort of surface area of that leaky gut, and the severity of it. So if only the smaller molecules are getting through, then it's not so severe. But if the larger molecules are getting through, then it's obviously more severe. So that's certainly used quite widely in research, from what I can see. There's also other ways. So fecal calprotectin is something that we test for fairly readily in general practice. And actually, fecal calprotectin is a measure of intestinal permeability related to inflammation of the gut. So certainly, people with inflammatory bowel conditions and celiac disease and things like that will actually have a degree of leaky gut, and that's been well accepted in the medical literature for a long time. And we can measure that by their levels of fecal calprotectin. So that can be used. And zonulin occlusins. So we've talked about zonulin because it's sort of something that is responsible for these tight junctions and sort of maintaining the barrier there. So if your zonula occludens is raised, then it increases gut wall permeability. And so that can be measured. But there is some debate about how good a measure that is in clinical practice. And I think there is a measure of that specific type of gut permeability related to those tight junctions. So it doesn't obviously measure the sort of damage to the mucus layer and various things like that. So that can be used. But again, all of these things really are just used in the laboratory and used for sort of research purposes. I spoke to my local labs, and currently in Glasgow, there isn't any availability of the mannitol and the zonulin tests for us to use. Obviously, fecal calprotectin can be used in those people with inflammatory bowel conditions, but it's not so useful in people without inflammatory bowel conditions. So we're a little bit stuck. So we can't really measure it very effectively. But that doesn't mean to say it doesn't exist just because we can't measure it. And I think, that's a debate, Siobhan, you and I have had over this time about, just because we can't measure it, does it mean it doesn't exist? So, Siobhan, what do you think about the journey from leaky gut to disease? How do we get there?
Okay, well, I think if you look at the two extremes, so if you have something like Crohn's disease or ulcerative colitis, where you've got sort of very large gaps and damage to the gut barrier, it's very easy to understand that you might get leakage of potentially harmful molecules such as this LPS that you've described. Now, just going back to this LPS, it's lipopolysaccharide, and it's a component of gram-negative bacteria of their outer cell membrane. And it's present, it's a structural element, but it's also there to protect the bacteria and help its survival. And it's particularly potent to human immune systems. So it's really easy to understand if you get a sudden acute surge of LPS getting into the body through this permeable membrane, you can understand that the immune system would have to mount a huge response, and that could lead to severe illness and fever and potentially septic shock. Now, that's obviously an extreme situation, but what we're talking about with these chronic diseases that we're suffering from in Western society is almost imperceptible, very low-grade leakage of part or all of these LPS molecules. They go into the lamina propria. And if you remember, I said at the beginning that you have 70% of your immune system situated here, ready to protect you from these sort of molecules. And it doesn't mount a huge response. You get this, what we call low-grade chronic inflammation that's meant to be key to the pathogenesis of many diseases. And I think this helps us understand, because lots of people say, well, wait a minute, gut health, how can the gut affect your mood? How could the gut affect your liver? How could the gut barrier dysfunction cause diabetes? But if you're talking the thing that's shared in the pathogenesis of all these diseases is this low-grade chronic inflammation. And obviously these inflammatory cells and cytokines, they travel through the circulation and message various other parts of the body, various other tissues and organs. And this is what connects gut health and gut permeability to systemic disease and chronic disease. And this is why it's thought to be so important. And also it explains why this very confusing thing for GPs when people come and say, could it be my gut? Could it be leaky gut? And their symptoms are very, very diverse, ranging from mood problems to gut problems to skin and joint problems. And I think this is what makes it so difficult without a clear diagnostic test. And Sheena, you've talked about the difficulties of testing. I think tests are going to be available over the next few years, and it will be something that's integrated into the NHS. But at the moment, we're in that difficult situation where people say, have I got leaky gut? We don't know because we can't diagnose it at the moment. But that doesn't stop us from giving very, very sensible lifestyle advice based on diet and various other lifestyle modalities that certainly are known from extensive research in many university departments in animal models and cell tissue models that we know do make a difference to the gut barrier function. And I think that's probably where we can be of assistance in helping patients traverse this sort of difficult area. So over to you, Sheena. What do you think?
Yeah, so you're right. So I think, how do we fix a leaky gut? Well, it all comes down to all of the pillars of lifestyle medicine again. But essentially, diet is one of the most important aspects of that because it has a direct impact on our barrier function. So whilst, as I've said before, that sort of high fat, high sugar diet will actually be very, very damaging to the gut wall. The opposite is true when you have a diet high in plants, high in fiber in particular. That's going to have a very beneficial effect on the gut wall. So fibers feed the colonocytes, they feed the bacteria, and they actually help us to maintain a much stronger, thicker barrier, and they help to the cells that produce the mucus. They help to bolster those cells that produce the mucus there as well. In addition, when we eat fiber, and these are fermented in our large intestine, there are a number of different short chain fatty acids produced. And one of these, butyrate, is particularly beneficial to the gut barrier. So if we have a nice high fiber diet, that's really going to protect it. There's lots of other nutrients that we can consume, polyphenols in lots of fruits and vegetables and things, which can have additional beneficial effects on our diet. Things like vinegar, it can be very beneficial to our gut barrier as well. So there's other things within diet that can be really, really beneficial. So that's really, worth considering if you want to have a really nice, strong gut barrier. And the bigger variety of plants in your diet seems to be associated with a better diversity and richness of beneficial bacteria and a more integral barrier. So that's really, really useful. So that's just one aspect. I think, obviously in this podcast, we will be covering, these type of things in later podcasts. And so what I'd like to move on to now is really some of the questions that we've had from people. So we've got one question that came in, which is from Mandy. She asked, what are archaea? And this came from our previous podcast where we mentioned archaea as some of the organisms that lived within the gut, alongside bacteria and viruses and various other things. So Shiv, what are archaea?
Yeah, well, when we did the course, Sheena and I actually had to go back to basics and learn about all these things. So most of the microorganisms in your gut are bacteria. And that's where most research is being focused, but more and more research is looking at other things like the protists and the fungi and viruses as well. But archaea are single-cell microorganisms, very similar to bacteria, no nucleus, and they were actually previously categorized as bacteria. They're kind of about the same size, they're kind of about the same shape. But some clever guy did genetic studies and found that they originated from a different genetic lineage. And they actually have quite specific genes and metabolism. These are meant to be sort of our oldest microorganisms. So they love extremes. So there's sort of wonderful stories about archaea being found in hot springs and salt lakes and deep in oceans and even in radioactive mines. So as far as we know, they are found in the human gut as well. We don't know any that are defined as pathogenic. We know that they do aid our digestion and they are specifically methane producing as well. But they can be considered as, about the same sort of in the same group as bacteria. But I think there's more research being done to try and find out exactly what they do in the human body. So there's certainly more research to be done there.
Fab. Right. So one more question, Siobhan. Who do you think is likely to have leaky gut?
Well, again, going from the numerous sort of scientific studies that have been done into animal models and some human models as well, it looks as if certainly anyone with a chronic disease, particularly obesity, diabetes, and inflammatory bowel disease as well, those are the people that are going to be probably going to have a very high risk of leaky gut. And even though we don't have a test to define that or diagnose that, it seems to me sensible in the sort of spirit of preventative medicine that we can still, everyone really should be looking at how to improve their gut barrier function. So although I couldn't say to a patient they definitely have leaky gut, I could say you are at high risk. And it would be sensible for you to look at lifestyle measures that will improve and bolster your gut function. And really, we should all be looking at this. We know there are things that wreck our gut barrier. So things like emulsifiers, high-fat Western diets, alcohol, stress, many medications such as non-steroidal anti-inflammatory drugs. And then we know lots of things that benefit and bolster the barrier. So fibre, prebiotics, polyphenols, having adequate zinc and vitamin D. So it seems really sensible for us all to adopt these things, which, as it happens, are also really good for other aspects of our general health as well. So I think everyone should be thinking about their gut health, but certainly those people who are at higher risk, I would be very confident in giving them this sort of advice or signposting them to good sources of advice about gut health. What about you, Sheena?
Yeah, I think, well, absolutely. I mean, this is sort of what we go on about all the time. But yeah, I completely agree, Siobhan. And, we just need to, I think, consider a leaky gut in anybody that really kind of presents with any disease. If we were to do a leaky gut test, if we had access to it, we would probably find that, a proportion of them didn't. But if a proportion of them have, and if they have other disease, we know they're likely to have dysbiosis as well. Then surely taking a view of trying to fix their gut is not a bad idea for most people. So yeah, I completely agree. So I think what we were going to do now is move on to a research article, one that's come out fairly recently. And I thought we'd take the opportunity to talk about emulsifiers because emulsifiers are these things used by the food industry, and they're used very widely in the food industry now. And I'm not convinced that they have been adequately researched prior to their use commencing, because they're really used very, very widely in a lot of things. So emulsifiers actually, they thicken things, if you think of it in that respect. So we use them to make oils into margarines, for example, or to make a very thin salad dressing into a thick salad dressing, or to bring fats and water together. So essentially, anything that's sort of been thickened artificially uses an emulsifier. And there's tons of different emulsifiers that we use, and a lot of them are plant derived, but actually, some of them appear to be a little bit more harmful than others. And so there was an interesting article that came out. It's called Direct Impact of Commonly Used Dietary Emulsifiers on Human Gut Microbiota. It's by Sabrina Naomi et al., and it was published in Microbiome in 2021. And essentially what they did were they not looking specifically at leaky gut, but looking at the impact of these dietary emulsifiers on just the microorganisms. So what they did was they used these really interesting things. So mini bioreactor arrays, which is essentially they take a freshly passed stool and they put it in an anaerobic chamber within 10 minutes of defecation, and then they use that to study the impact of emulsifiers on that gut microbiota within that ecosystem. So they looked at 20 different emulsifiers. So from the xanthan gums to the carrageenans, which are derived from seaweed, maltodextrins and sorbitans, glyceryl sterates, all these sort of really fancy names of things. And they saw what they did in these situations. So interestingly, so the xanthan gums and the sorbitol and the glyceryl sterate, these ones actually increased LPS. Now, we've already heard that that's pretty damaging for our gut wall, and that became quite a significant increase in LPS levels. The carrageenans and the gums actually increase the virulence of some bacteria. So this is quite interesting because bacteria like E. coli actually have these little hairs protruding from them. They're called flagellae. And what happens when the emulsifiers come into contact with them is it makes these hairs super sticky. And it makes them become more toxic to us, because if these bacteria are super sticky, they can adhere to walls and they can create disease more readily. And this is certainly the case with E. coli. So there was this sort of increased virulence of bacteria like E. coli in contact with the emulsifiers. There's a variety of these emulsifiers that increase bacterial load. And it was worth noting that a few of them didn't have a great deal of impact. So soy lecithin and mono and diglycerides didn't have a huge amount of impact on the gut microbiota, which is reassuring. But what was slightly more concerning was that some of them actually continue to have a harmful effect even once the emulsifiers were removed. So it seemed to be quite a long-lasting impact from the emulsifiers, which is a little bit concerning that they can continue to cause you harm. It's also worth noting out with this research paper that there are other papers as well that have talked about what emulsifiers do to your mucous membrane. So the mucous lining of the gut wall actually dissolves with emulsifiers because it's very sort of mucolytic. And that can lead to sort of, gaps in the mucous lining and increase the likelihood of inflammatory damage to the wall itself. So it does. There have been studies that have revealed that leaky gut does happen as a result of emulsifier contact. So it's worth. I just wanted to outline that little research article because I thought it was quite an interesting one, and because we all probably consume emulsifiers fairly regularly, and I think there is actually a dose response with them. So the more emulsifiers you do consume, the greater the impact from them. And as I say, they're all slightly different, but they do appear to cause a lot of harm there. So yeah, what do you think, Shiv?
Yeah, well, it is very interesting. It's important for people to remember that these things, which are assignated as GRAS, which is generally regarded as safe, which is the American terminology. So all these additives and emulsifiers, and I think there's over, it depends how you grade them in different countries, grade them in different ways, but I think there's over 100 emulsifiers available. And they were meant to be helpful to mankind. They were meant to increase shelf life and texture and stop separation and make it look nicer and feel nicer in our mouths. So there are numerous things that these emulsifiers do, but they were brought out pre-gut microbiota and before we understood the complexities of the gut barrier, I suppose. And certainly in animal studies, and particularly mouse studies, it's very clear that many, not just one or two, but many of these emulsifiers show within a very short time increased inflammation, can produce colitis in mouse models, increased permeability using some of these measures that you've discussed. And also they alter the gut microbiota composition. And your gut microbes are very important in terms of they help produce mucus and this butyrate they produce, which is very important for the gut barrier and to feed the gut cells. So in all sorts of ways, they really kind of wreck or cause a lot of damage to the gut barrier. And that's why they're associated with diabetes and obesity and metabolic syndrome. So it's kind of, how do we go back? Because they're already available and ubiquitous in many of our foods. And we've got lots of scientists who are studying particularly emulsifiers, who are sort of shouting into the void that they are worried about what they're doing to our gut barrier and that they may be part of the issue with ultra-processed foods, which we know for probably a variety of reasons are really intricately involved in the pathogenesis of many of these chronic diseases. And this is another bit of the puzzle. But really, it's kind of no one's really listening. But it is a concern, and they are asking people to do research before we allow this stuff to be put into our food. But of course, it's already in our food. So how do we take it out of our food? And then you've got kind of big food and all this sort of the various financial interests that it's not in their interest to sort of look at this aspect of our what's going into ultra-processed food. So, yeah, it is a concern. And that's why this move towards whole food and, trying to avoid additives and emulsifiers when you can is a very sensible way to go.
Yeah, and as a GP, this is something I've had a bit of success with in the last year with patients who are having, for example, recurrent UTIs. We know E. coli is a common causer of UTIs, and actually I've taken a number of my patients who are in this situation off emulsifiers and increased the fibre in their diet, and it has actually resolved their recurrent UTIs. So I've actually had some success in removing these from people's diet and helping reduce disease. So that's a little anecdotal thing at the end there. So Siobhan, we were just going to summarise 3 basic take-home messages really from today's podcast.
Yeah, so I am a huge fan, as you can probably tell, of the gut barrier. So I just want to remind everyone that's listening that your gut barrier, you have one cell thick, your epithelium with these intricate junctions that open and close. Then you have your mucus on top of that and your microbes. You have your immune system. 70% of your immune system sits in your gut. And then you have your enteric nervous system, which is these wonderful nerve fibers. So it's a really complex, wonderful structure that we need to look after. And we know that things that are good for it and we know that things that are bad for it. So, really focusing on this area will, I think, be a huge part of improving people's health.
Yes. And we can absolutely say that leaky gut does exist. And it is associated with numerous diseases. And there are stacks and stacks of literature on this if you want to have a wee look. Siobhan and I, as we've said, we've literally read hundreds of articles and research papers for our course that we've written. And during that we've come across so many articles on leaky gut and so many references from major microbiome researchers and scientists on increased gut permeability and leaky gut, and they use the term leaky gut very frequently. So this really does exist, and this is very important that we all know about it. So I would urge everybody out there who hasn't heard of it to look more into this. Okay, Siobhan, do you want to finish up and let us know what's happening in the next podcast?
Yes, well, we have got lots of requests for lots of different subjects that people want us to look into, but one of the favourites seems to be fermented foods. So we thought we'd kind of have a practical look at fermented foods, and we're hoping to get someone who's got a special interest in this area to come and help us do our next podcast. Sheena, can you tell us a little bit more about that?
Yeah, so I'm hoping to bring on a really amazing lady called Janice Klein, who is she's a real specialist in fermentations, and she's locally known as the fermentation queen in Scotland. So hopefully we'll get her on next time, and we'll have a nice in-depth chat about fermented foods, what they are, why we should know about them, why we should use them and eat them, and have them regularly within our diet. So I think that's going to be really interesting. And I'm really looking forward to picking her brains, actually, because, it is such an interesting subject. So we'll go. Thank you so much for listening today. I hope you've enjoyed this podcast. And if you have, then please remember to take a wee look at the gut microbiota for physicians. It's an online course that Siobhan and I have written at the BSLM website. So have a wee look at that, and we'll speak to you next time. Bye.
Bye.